We've said that enzymes are capable of catalyzing metabolic reactions. And basically what that means to us is that enzymes are going to make these reactions occur more readily. In more technical terms, that means they are increasing the rate of the reaction. The reaction will produce product more quickly. Now, on this slide, there's a very complicated looking graph, and I'm going to walk through this graph with you to point out what we need to take from this particular illustration. On the x-axis here is the progress of a chemical reaction, and on the y-axis is what they're listing as free energy. Think of free energy as just being the energy that's available to a cell to do some work. Remember, when you break apart molecules, that's catabolic metabolism, and that releases energy. When you build molecules, that's anabolic metabolism, and that requires energy or consumes energy. So what exactly is happening with the enzyme in terms of all this energy? In this particular graph, we're shown a couple of reactant molecules, something called A, and then something else called BC. These are our substrates, or our reactants. You can see over on the other side of the graph, after some time has gone by, that we now have product molecules, something called AB and something called C. So in order to go from these substrates to these products, what had to have happened is that the bond between BC must have been broken, and a new bond between AB must have been built. So very classic sort of metabolism going on here. We have a catabolic breaking and an anabolic building. We have to then determine overall, is this an anabolic reaction or a catabolic reaction? Well, that has to do with whether or not overall energy was consumed or energy was released. I'm not going to get into too much of the detail of how we do that mathematically. Just understand that in any complex metabolic reaction, if overall energy had to be pulled into the reaction to make it happen, we call that endergonic or anabolic. Overall, if energy was released from the reaction, we call that catabolic or exergonic. Now, where does the enzyme come in? I want you to follow this line here where the substrates are, and notice the dotted line. Notice that the free energy, the variable on the y-axis, is going up, 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 up until we hit something called the transition state. Then it goes back down, 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 down till we get to the product molecules. This transition state is a place, if you will, during the reaction where we're at a very high level of energy. If I trace the peak of this dotted line over to the y-axis, I'm at a very high level of energy, free energy. Think of the transition state as the time during a chemical reaction where all the old bonds have been broken down, but none of the new bonds have been built yet. It's an intermediate state. It's also a time of a very high energy level, the transition state. Now, if this reaction were to occur without an enzyme, it would not be easy for the cell to hit this state, to hit this level of high energy. We in fact give that level of high energy a name. The energy that's required to hit this transition state is called activation energy. We shorthand that as EA. So during this reaction, without an enzyme around, it's not easy to hit the transition state. It requires a lot of activation energy. Once you bring an enzyme in, though, something very interesting happens. Once you bring an enzyme in, the peak level to get to the transition state has been reduced. In other words, enzymes lower the activation energy, or the energy required to reach the transition state. Enzymes make it easier for this reaction to go forward. It's easier to achieve this transition state, this intermediate place, before products get formed. So, to summarize, the activation energy is the energy that's needed to reach the transition state of a metabolic reaction. That place, that time halfway between having a substrate molecule and having a product molecule. Without an enzyme around, activation energy is pretty high and it's hard for the cell to reach it. But with an enzyme around, the activation energy gets reduced or lowered, so it's easier to reach. This is how enzymes increase the rate of a chemical reaction. Now, enzymes are very powerful things. They can make chemical reactions go faster. So it's in the best interest of a microbial cell or any kind of cell to pay close attention to their enzymes. You wouldn't want to let enzymes just do these reactions all day long without some control in place. Enzymes, therefore, get regulated by the cell. Cells have to have control over them, in other words. Now, there are two broad types of control I want us to be familiar with. One of them is called competitive inhibition, and the other is called allosteric inhibition. Let's look at competitive inhibition first. During competitive inhibition, a molecule that looks a lot like the substrate molecule is going to be used by the cell to control the enzyme. This molecule is very similar in the size, very similar in the shape of the substrate, but it's not the substrate. And what it means is that this competitive molecule is going to be able to bind to the active site. Remember, the active site is specifically shaped for that substrate. And this competitor is going to come in and basically pretend to be the substrate and bind to the active site. By sitting in the active site, the competitor molecule is blocking the substrate from coming in and sitting there. And because the competitor is not the actual substrate, the enzyme can't do anything with it. It can't make any product with it. So during competitive inhibition, we have essentially turned off the enzyme temporarily by blocking the active site from the substrate. Take a look at the drawing at the bottom of the slide. What's drawn in green represents an enzyme. And you can see that there are these sort of hollowed out areas where these substrate molecules can come in and bind. Remember, the substrates are shaped so that they fit perfectly in the active site. Now, if the cell wants to take some control over this enzyme, one of the ways it can do that is by producing a molecule, shown in blue, that looks very similar to the substrate in its size and its shape. The competitor molecule can come in and actually sit in the active site and block the actual substrate from coming in. The competitor molecule is not substrate, so the enzyme can't do anything with it. It can't make product with it. So at least temporarily, this enzyme has been turned off. The second kind of inhibition I want us to be familiar with is called allosteric inhibition. This one is similar, but just a little bit different. In allosteric inhibition, there's going to be another inhibitor molecule, but this one doesn't bind to the active site. It binds somewhere else. when it binds to the enzyme at a location other than the active site, it's going to cause the enzyme to fold a little bit differently. Remember, anytime you stick any molecule onto a protein, you're going to cause that protein to fold just a little bit differently. Enzymes are no exception. So when an allosteric inhibitor binds to an enzyme at a location other than the active site, it's going to cause the enzyme to fold just a little bit differently. And that refolding is going to change the shape of the active site. That's what they're showing us down here in this drawing. Now, they're showing us two kinds of allosteric regulation, but we're particularly interested in this one on the right. Allosteric inhibition or allosteric deactivation. The regulatory molecule is drawn as this blue triangle. It's going to come in and it's going to bind to the enzyme at a spot other than the active site. When it binds there, it's going to cause the enzyme to change shape. And what they're showing us is that the active site is not shaped quite as it was over here. The hollowed out areas where the substrate can come in and bind have become a little shallower. They've just changed enough so that the substrate can no longer fit perfectly in that active site. The substrate can't bind now because the active site has changed shape. As long as this regulatory molecule is sitting here, this enzyme is inhibited. Now, in addition to the cell being able to regulate enzymes, environmental conditions also influence how active or inactive an enzyme will be. There are two very important environmental conditions that will affect how active or inactive enzymes will be in a cell: temperature and pH. Now, temperature is really a measure of kinetic energy. We talked a little bit about this in class. It's a measure of how molecules are moving, how they're vibrating, if you will. When you alter the temperature around a microbial cell, you're altering how things are moving inside that cell. That includes both the substrate and the enzyme. So, if the temperature goes down, the molecules inside the cell are moving a little bit less. And if we took the enzyme out of the picture, it would mean that molecules are colliding with each other less often, and chemical reactions are occurring less frequently. When you put an enzyme into the picture, it means that the substrate is colliding with the active site less often. So we have enzymatic catalysis going on less often. The opposite is true as you increase temperature. As molecules move more, it's more likely that a substrate would bump into or collide with the active site of an enzyme. So you would see reactions occur more frequently. Now, there are obvious limits to this. If the environment around a microbial cell gets too cold or too hot, the enzyme itself is going to be affected. We're going to see the enzyme actually unable to perform at all. So there are limits to these changes in temperature in terms of how well an enzyme can perform its job. With pH, something very different is happening. As we alter the pH around a microbial cell, what we're changing is the amount of hydrogen ion that is in the solution of that cell. Hydrogen ion, remember, becomes more plentiful as pH drops down. As the pH number gets lower and lower and lower, we're saying that the solution is getting more acidic and there's more free hydrogen ion around. As pH levels go higher and higher, we say that the solution gets more basic and there's less free hydrogen ion around. So at a nice neutral pH of 7 or so, the enzyme is sitting in a bath of hydrogen ions that are just right. And there's a certain number of hydrogen ions binding to the enzyme and influencing the shape of the enzyme, and everything is just right. As you start to alter the amount of hydrogen ion, though, you're going to alter the shape of the enzyme, because hydrogen ions will bind to an enzyme and cause it to change shape. Just like anything that binds to a protein will cause a protein to change shape. So, all enzymes in all microbes, and in fact in all cells, all enzymes have an optimal temperature for their best performance. All enzymes have an optimal pH for their best performance. It doesn't mean that they can only work at that optimal temperature. It means that they work optimally at that optimal temperature. They work best at that optimal temperature, and if the temperature goes a little bit down or a little bit up, they'll still function, but they'll function less optimally. They have an optimal pH for performance, so if pH goes down a little bit, more acidic, or up a little bit, more basic, they'll still function, but they'll function less optimally. Take a look at this graph. On the x-axis we have temperature in centigrade, and on the y-axis we have relative activity, a percentage of enzyme activity. Now there are two different lines on this graph, two different data sets. What we're looking at is one kind of enzyme called glucose 6-phosphatase and how it performs inside two different kinds of bacteria. In one of the bacterial cells, this enzyme is functioning at a particular optimal temperature. If you look at this yellow line, you can see that at this point right here, the glucose 6-phosphatase is at 100% activity. And if we trace that down to the x-axis, we see it's right around 40 degrees centigrade. This happens to be the body temperature of a human being. This bacterium, in fact, lives inside a human being, a human body. Maybe it's in the intestinal tract. So it has evolved to become adapted to human body temperature. And its enzymes, including this glucose 6-phosphatase enzyme, has evolved to function best at this body temperature. Now, it doesn't mean it can only work at that body temperature. If we drop the temperature down a little bit this way, or if we raise the temperature up a little bit this way, we're still making product. We're just seeing the relative activity drop off. Maybe the enzyme is only 60% effective, or 75% effective in producing product. Now look over here at the red line. This line represents the activity of a glucose 6-phosphatase enzyme that was taken out of a bacterial cell that happens to live in hot springs out in the environment. So this bacterium has evolved so that its enzymes function at 100% at a much higher temperature, more like 90 degrees centigrade. Those enzymes are going to produce product the best, optimally, at 90 degrees centigrade. They will still be able to produce product at lower temperatures. They'll still be able to produce product at higher temperatures to a point. It's just that they're producing product less optimally. The enzyme is functioning at maybe only 50%. Or maybe it's functioning with a relative activity of 75%. Still making product, just not making product as well.